Good morning. There'll be, I'm going to do this in probably one or two segments because I'm home, and depending how the dog permits me to do that. But this is the conclusion of the immune system. I know it's a very difficult thing, and all of these different co-stimulators and cytokines that are there. I mean, you can take a look at this yourselves. I'm not going to go crazy over it from a test perspective. But again, this is the idea that the T cells, particularly, are extraordinarily powerful. And they generate a tremendous amount of what we call interleukins. And the interleukins, unfortunately, are both good and bad. And I has acquainted you a little bit with some of the things that I have taken and potential damage that they can do, as well as the potential benefits that are there. So, again, you're going to see the video about the antigen binding and co-stimulation. If the dog barks, we'll start over. And a variety. But again, this whole idea of double recognition, specific receptors, we're not going to go into. And as you can see, if the system is working properly, it will peak within a week, this response. And that's a very, very efficient way of dealing with any variety of intracellular or cellular alterations that are there. And if it works properly with regard to T cells, as you will see, they'll undergo apoptosis. They'll be eliminated over the next 7 to 30 days. And it's all about the cytokines. That's why you have to have this idea of regulatory T cells, what we used to call suppressor T cells. If not, as you can see, the stimulating factors can lead to really effectively cancerous. It just causes mitosis to go crazy. And yes, there'll be memory cells. So they're demonstrating you, and I'm not going to go through all these prompts. It's a little bit easier if I show it to you with the animation. The chemical messengers, again, typically referred to as ILs or interleukins, and interferon is part of both the innate and various other types of interferons, particularly with regard to viral infections, are because we just don't have one kind of interferon, are part of this interleukin release as well. The big deal is interleukin 1, which is released by T cells, stimulates additional release of interleukin 2. Interleukin 2 is that compound that comes from that helper T cell more than any place else. And as you can see, that will even additionally synthesize more receptors on other cells. So to make other cells more sensitive to interleukin 2. It's the big player, as you can see. Encourages all other cells in the line to grow more regularly, amplifies it, regulates it. Okay. And then other innate, like the gamma interferon, which is effectively, it will also have enhanced killing power. And you can see this whole laundry list that we kind of looked at ever so briefly before, these selected cytokines. And they're really nasty. So we take a look at here's the interferons, many cells, antiviral effects, NK cells. This is the one from lymphocytes, particularly in the face of a viral infection that hardens the other cells against it. This is interleukin 1 is pro-inflammatory and works for T cell activation. But here, from the helper T cell, that T sub H, that's really the big player in all of this. And you can see there's a lot of these. And because these are effectively proteins, they've been isolated, they've been understood, we can duplicate them. And by the same token, we can fight against them. And that's what I meant by some of the medications I've taken. The interleukin 17 inhibitor, okay. So it's a pro-inflammatory. So if you block it, it acts as an anti-inflammatory. I was taking that for arthritis, but there's a lot of side effects that are associated with these things. Okay, so the helper T cell is the big player in this. Activates both the humoral, the antibodies, and the T cell arm. Okay, once it's primed, it activates all other cells in the list, as you can see, and without it, there's no immunological response. And I should show it to you than go through again all of these different things, which are you can see the complexity. Helper T cells mobilizing both humoral and cellular. So here you have between the antigen presenting cell, the helper T cell, and the killer T cell, all involved in this elaborate activity. And then you can take a look at these subsets of helper T cells. So there's more than this. So different subsets. This plays a role with most cellular immunity. This mobilizes eosinophils, which help to release chemicals that sort of gobble up or break down worms, which are multicellular. This is that big, the link between the adaptive, and that's the one that they were trying to limit because of its overactivity is the source of a lot of immune diseases, at least it's believed. And so what happens? Virus-infected cells, intracellular bacteria, intracellular parasites, cancer cells, transfused, transplanted cells. Again, remember all those. Again, they poke holes. Those are perforins. They release powerful enzymes that stimulate apoptosis. So here, the perforins are there, the granzymes. Here's the target cell in yellow. Here you can see the release of these first perforating proteins, poke little holes, and then the enzymes that go through called granzymes that activate the cell's apoptosis. And this is in action in a real micrograph. You can see the T cell attached to this. And cancer cells are very irregular. You can hardly recognize them, and it latches onto it, and boom, there it goes. So, oops, beg your pardon. Natural killer cells recognize other signs of abnormality that killer T cells don't look for. That they don't have those MHC type 1s, antibodies coating the cell, etc. Surface markers. So when you see in the immune system on a peripheral smear, if you look at any white blood cell, any blood smear, and you see lymphocytes, which we looked at a little bit in the blood unit, the lymphocytes are either natural killer or cytotoxic C cells that are everywhere. They're even ones that were activated and still prowling around. They are still part of your immune system. And then the regulatory T cells, they slow things down. So you can inhibit the response with IL-10 or transforming growth factor. So there's different types of things that are there. So they suppress the self-reactive ones. And again, trying to use a lot of these regulatory cells and slowing down the response, let's say, to transplants or to more sophisticated techniques is something that's it's sort of in the working. And we're just seeing the early parts of it. Let's see if this will play nicely. Sorry about that. There we go. I'll try again.
This patient's cold is caused by rhinoviruses, shown here in red, that entered her nasal passages and escaped the first-line innate defenses such as mucus and cilia. Some of these rhinoviruses entered the respiratory epithelial cells where they replicated, releasing more rhinoviruses into the patient's nasal passages and tissue fluids. In the tissue fluids, cells of the second line of innate defense, such as this dendritic cell, carry out surveillance for anything that appears foreign. Dendritic cells engulf the rhinovirus within an endosome. The endosome fuses with a lysosome, whose enzymes digest the rhinovirus. The resulting rhinovirus fragments are called epitopes. The dendritic cell then attaches the rhinovirus epitopes to presentation molecules called MHC2. This complex folds outward so that it sits on the outside of the dendritic cell membrane. The dendritic cell then enters a lymphatic vessel and travels to a nearby lymph node. Here, the dendritic cell, part of the innate defenses, encounters a helper T cell, which is part of the adaptive defenses. The T cell receptor, or TCR, of this helper T cell recognizes the rhinovirus epitope, while the CD4 glycoprotein associated with the TCR recognizes the MHC2. The dendritic cell then stimulates activation of this helper T cell. Adaptive defenses help to control the current infection, but also provide immunity against future infections by the same rhinovirus. They are carried out by two types of lymphocytes, B cells and T cells. Immature.
Very quickly, rhinoviruses, which they depict here as activating T cells, rarely do. They're mostly B cell. That's the common cold. So that's typically not intracellular. There may be some exceptions, but I just wanted to, but they're using it for example.
B cells conduct surveillance in the lymph nodes, using B cell receptors, or BCR, to recognize only one specific epitope. The BCR on the left does not recognize the rhinovirus, but the BCR on the right does, and this B cell is selected for activation in a step called clonal selection. The selected B cell takes in the rhinovirus, degrades it, and presents the rhinovirus epitope with its MHC2. Our mature helper T cell recognizes the rhinovirus epitope with its TCR and the MHC2 with its CD4. Then the helper T cell secretes chemical messengers known as cytokines that activate the B cell. The B cell proliferates and differentiates in a process called clonal expansion, resulting in two types of cell. Memory B cells carry the memory of this pathogen for many years. Plasma cells secrete antibodies that conduct surveillance for more rhinoviruses, grouping them together so they are easy targets for phagocytes. Antibodies in the blood and tissue fluids seek out free rhinoviruses. The antibodies neutralize the rhinovirus by binding to its surface molecules, thus preventing attachment to host cells. Antibodies are humoral defenses that target free pathogens. But what about the rhinoviruses that are already inside our patient's cells? Another type of adaptive defense cell, called a cytotoxic T cell, can locate and destroy rhinovirus-infected cells. It provides cell-mediated, or cellular, immunity. Let's return to our patient's lymph node to see how cytotoxic T cells become activated. The T cell receptor, or TCR, of this particular cytotoxic T cell is genetically programmed to recognize the rhinovirus epitope when presented by the dendritic cell. This time, the epitope is presented differently, with an MHC1, which is recognized by a CD8 glycoprotein on the cytotoxic T cell. With the help of cytokines from a nearby helper T cell, the cytotoxic T cell is activated and clonal expansion occurs, resulting in some long-lasting memory cells ready for a subsequent exposure, and many active cytotoxic T cells, or CTLs, that leave the patient's lymph vessel to search for infected cells that present rhinovirus epitope with their MHC1. The epitope is recognized by the TCR of the cytotoxic T cell, and the MHC1 is recognized by the T cell's CD8 glycoprotein. The cytotoxic T cell then secretes perforin molecules, which form channels in the infected cell's membrane. Granzyme, also from the T cell, passes through the perforin channels and activates enzymes that lead to apoptosis, the programmed death of the infected cell. The cytotoxic T cell then moves on to look for another infected cell. Together, the humoral and cellular defenses are the body's third line of defense in fighting a very specific pathogen. The humoral defenses ultimately in the form of antibodies, conduct surveillance for rhinoviruses that are outside the body cells, in the patient's tissue spaces and bloodstream. The cellular defenses, in the form of cytotoxic T cells, search for rhinovirus-infected cells, inducing apoptosis when they are found. Memory T and B cells help our patient's body to recognize and more rapidly fight the same rhinovirus should it infect her again.
So understand that's basically, even though rhinoviruses don't generate teeth, but that's trying to give you the complexity of the system, and understand that the memory cells are important, and the regulatory B cells, particularly, or beg your pardon, regulatory T cells like the suppressor T cells are really another important arm for this. So we have this on our PowerPoints. You can listen to this again. It's more to give you an understanding than go into co-stimulatory and regulatory and what binds to what. Again, CD4s, helper T, CD8s, cytotoxic, or what we call killer T cells. And more importantly is what they're attacking. If it's a cancer cell, it's a killer T cell. If it's a common cold that's just floating around and not inside of the cell, we're looking at antibodies. We're looking at B cells. That's the idea. Now, this is a little bit more about, that's pretty much how the immune system works. These are just some specifics. So when you hear about somebody getting an organ transplant, like getting a kidney, it's not their kidney. That comes from the same species. That's called an allograft. And so we have to, in order to see if they're compatible, we look at certainly the blood type and blood groups and all of this combination of MHC antigens. And you might remember me saying it before, we'll do a screening. And let's say there's 100 markers they're looking for in people with the same blood type. And what they'll do is if they have 75, then there's sufficient amount of compatibility that kidney, let's say, would take. And obviously we continue to use immune suppressants, and that's another catch-22. And so the immunosuppressants are always there. So it's not easy. It's a little bit easier if you have a very strong compatibility donor, like a sibling or a parent or a child, that type of a thing. There's a lot to it, and transplant science has improved tremendously. Okay. So the big problem is infection, because your immune system is suppressed. Okay. And I mean, that's optimal. And this book was written and this PowerPoint put together maybe 6 or 7 years ago by this time. So probably the numbers have improved a little bit, but we call that a survival rate. So a 10-year survival rate of 50%, at least at that time, was good. So it means half the people survive after 10 years. And there are plenty of people with longer things like that. And now we get into these different problems that are there without getting into the diseases just let. Immunodeficiency. And this is very famously, and it's changed a bit now. So congenital or acquired. When we look at the congenital, what you're born with, okay, this is called SCID, or severe combined immunodeficiency syndrome. We used to call it the bubble boy syndrome, because the deficit in B and T cells in this particular enzyme that are lethal to these cells. And nowadays we can do bone marrow transplants and have improved that. That was a very famous, and interestingly, they've portrayed that in the movies on more than one occasion historically, where they could have no contact with anybody from the outside. They basically had to exist, sequestered from anybody else, and almost anything that potentially had a pathogen, a disease-causing agent, could kill them. Acquired, okay, Hodgkin's, talked about that. Maybe I mentioned it previously, but I usually do with lymph nodes, with Mario Lemieux, is a B cell cancer, and again, immunodeficiency. So it's fortunately there's aspects of it that are more treatable than others depending on the severity. So those are just some examples. Now the most famous, at least today, I think, of the acquired ones is AIDS. Now AIDS stands for acquired immune deficiency syndrome. It is caused by HIV, the human immunodeficiency virus. And what it does basically is destroys the helper T cell population, and particularly attaches to that CD4 marker, and along with helper T cells and other macrophages. And in its late stages, once the helper T cell population falls below a critical level, your immune system collapses. And at that point, they're going to die of an infection. And there's going to be a lot of other things that occur. They become basically, it's like a cancer eating them up inside. But the reality is their immune system is just can't fight typical things. And we begin to see and this is very characteristic of long-term chronic diseases: weight loss, night sweats, swollen lymph nodes. I mean, if you want to see its severity, take a look at Freddie Mercury, and this is before we had any realistic treatment. Freddie Mercury in the early Queen days, and there's plenty of videos of his last performance. And I think the song is "Those Were the Days," and he died shortly thereafter. And you could see a shell of what he was. He was in terrible pain when he was doing it. And what effectively defined it, when we recognized it, was this: an opportunistic infection. Infections, that means that you don't get unless your immune system is collapsed. He had Kaposi's, but back in 1981 in San Francisco, where they had a large gay population, and anal sex is a very easy way to transmit this because the anal mucosa is more fragile, and as opposed to the vaginal mucosa. So gay male anal intercourse was a very convenient way to spread this in a population that where typically they did not use anything like condoms or birth control because they obviously weren't in a birth situation. So it really was facilitating the spread more than anything else. And this physician noticed an increase in a particularly unusual pneumonia called pneumocystic pneumonia, known as PCP for pneumocystic pneumonia. Now it's called Jirovecii, pneumocystis jirovecii. It's a fungus. And our bodies reject fungal pneumonias very easily. We have a very good surveillance system for that. It's very rare, unless you're dealing in the Southwest when someone gets valley fever, because there's just an overwhelming amount of fungal spores at certain times in the desert area. And I talk about that a lot in Micro. So we know that the HIV, what happens over time if someone's not treated, that virus proliferates, and even some of the T cells become little factories that give off the virus. That's the problem. As they collapse, they become little factories that make the virus and release it. And they go throughout all body fluids. And so blood transfusions, needles, the usual thing. This is typical blood to body fluid, and certainly mother to fetus, which is something that treatment with anti-HIV antivirals have been very successful in trying, because before a mother who was untreated, that baby was going to have HIV and was going to die of AIDS. And so all of this is part of it. That's there. The typical time without treatment, somewhere between 2 and 10 years for the immune system to collapse, depending on the health of the person, their lifestyle, a variety of other factors. It can be much sooner than that. And so someone who's had it for a long time, that immune system collapses, and it's just everywhere. And there's, you can Google and you'll see MRIs of AIDS dementia, and you can see the difference between an MRI of a normal brain and an AIDS brain. Oh my goodness. And so, and again, it's attached to the CD4. If you did not know and you haven't had Micro, I understand, most of you, it's called RNA reverse transcriptase. It's a very interesting virus. It rewrites. It's an RNA virus, like COVID, that when it enters the cell has an enzyme, COVID does not, that basically rewrites the RNA into DNA, reverses transcription, that's where it gets its name, and then reprograms the cell. We actually use that in gene therapy in its early days now, of trying to reprogram cells so they can be better. So that's where we're actually taking the bad stuff out of the virus and trying to use it for good, which we do a lot with things. Okay. Lots and lots. The problem with RNA becoming DNA, it doesn't have the same proofreading mechanism that your cells and my cells have. Make from DNA making RNA, or our cells undergoing mitosis. There's a lot of proofreading that goes on that allows our cells, that basically if there's an error, they destroy the cell. We don't have that. So that's why there's a lot of problems associated with it. I go through chapter and verse in this. Okay. So there's no cure, but now basically it used to be a triple cocktail. Now it's a four-way cocktail in combination is the standard treatment is because they've developed more and more medications. There weren't a lot of antivirals, but in 1988 there was something called the Orphan Drug Act, and has nothing to do with someone being orphaned. It has to do with financial incentives and getting ready to distribute incentives for pharmaceutical products in underserved populations. In other words, there aren't that many people who had HIV/AIDS, and it was a financial incentive. And this was brought on by the Hollywood and the entertainment community because very prominent people started to have this, and a lot of money came from big people in Hollywood, as it's apt to do for certain causes, both medically, politically, environmentally, whatever. And that's a whole other discussion. It's not part of our class, but understand that they basically said, hey, you want my support and my financial backing, you need to make a bill. And they did. Thankfully, it was a good bill, for this, because Liberace was a very famous entertainer who was effectively a closeted gay man, and Rock Hudson, who was an action hero and a male, basically, he was an actor, everybody was super handsome and virile and tough guy. Boom. People didn't know that again, he was a closeted gay man at the time. So and that was the start, and there are many others that are there. Autoimmune diseases, I know I have a variation of one. I'm fortunate that I'm able to do what I'm able to do because it's kept under control by medications, and thankfully it didn't trouble me until considerably later. So immune system loses the ability to distinguish self from foreign. Effectively, that's it. So your immune system starts viewing normal stuff as foreign, if you want to think of it that way. And production of antibodies and sensitized cells, actually killer T cells, actually attack your healthy tissue. So the antibodies can attack it. We see that in streptococcal infection. So an untreated or undertreated strep throat will lead to something called rheumatic fever, which affects both the joints and the lining cells of the heart. Boom. That's an autoantibody. And then and a lot of these where we have T cells kind of gone crazy. So rheumatoid arthritis, and it does more, yes, its primary area is destroying joints, but it is extraordinarily nasty and will just, in certain individuals, it'll shorten their life immediately. About 10% of the people don't survive very long after the diagnosis. And on the other hand, about 10% of the people just carry the diagnosis. And there's a lot of them that are like that. Multiple sclerosis is like that. Okay. Myasthenia gravis, to some extent, is like that. Most of the time, it's a bell curve on how people are affected. About 10% of the people are diagnosed with it, but it doesn't limit them, but they do maybe have some minor treatment that's involved. But it doesn't affect them from doing their job or living a normal life, and 10% of the people succumb early. In between that bell curve, those are two standard deviations from the mean, and then you have the mid-range. About 40% of the people are affected, something like I would have to take medications, have good days and bad days, but they can do basically everything else. I have no problem with what's called ADLs, activities of daily living, getting dressed, taking a shower, going to the bathroom, etc., driving a car, all that good stuff. The bad side of the 40% are people who are affected by ADLs. In other words, they can't do those. That's what if you're interested in occupational therapy, that's your job. Improvement with activities of daily living. And that's why they do so much upper extremity. So people can write, people can use, even if they can't walk, they can go and use the kitchen, they can bathe and shower with a certain degree of assistive devices, and they're improved amongst those things. So about half the people have significant impairment, and particularly so of activities of daily living, and half the people do not. That's typically the bell curve with this. So myasthenia gravis, it affects what's called the neuromuscular junction, which you probably learned about in AP1. Rheumatoid arthritis, it's not just joints, it's all over the place. Nodules that form, suppressed immunological function, all of those things. MS, very severe. It's a demyelinating disease, so the myelin sheath degenerates both centrally and peripherally. This one involves attacking the thyroid and produces large amount, because the thyroid kind of works almost backwards. With this particular one, it actually makes more rather than making less. But we see a lot of evidence of antigen antibody complexes, which are part of autoimmune phenomena in diabetics. They form in the kidney and they clobber kidney function, and we typically see it in type 1 diabetes. That's why the feeling is now that type 1 has a lot to do with autoimmune phenomena, that maybe there was some type of viral intracellular phenomena could possibly have created this problem. There's a lot of theories behind it, but those antigen antibody complexes in the kidney that damage kidney function, and we'll talk about that more in the kidney. Lupus, again, antigen antibody complexes that form not just in the kidney, but all over multiple organs, and the glomerulonephritis is the one in the kidney that's more vis-a-vis the type 1 diabetes. So typically, you can use anti-inflammatories. In the old days, all we had was steroids, and a medicine that I take called sulfasalazine, which we still don't know how it works. I take it now, and we still don't know how it works, but it helps. And that was the limited way people with rheumatoid or psoriatic arthritis or lupus. It was a cocktail. I did a very interesting student, what we called an externship. It's like I was in between my 3rd and 4th years of medical school, and I tagged along at the Medical College of Virginia in the Vets Hospital there with an eminent rheumatologist, real famous. I was fortunate enough to work with him for a couple of weeks out of the 2 months that I was there, and we were treating rheumatoid arthritis. He had a ton of patients. And it was like, I asked him, how do you figure out what to treat him? He goes, it's like a recipe. He says, we give them muscle relaxers, we give them anti-inflammatories, we give them pain pills. At the time, we didn't have the sophisticated stuff that I like taking today. These, ones that interfere with interleukins. We didn't, with genetic engineering and genetic medicine was non-existent. We didn't have the capabilities of even making insulin, which didn't start until 1977. This was 1976. And what happened? He was using gold. Gold is an old-time treatment. It's called myocrysine. He would inject them with gold in an effort that the heavy metals were a suppressant immunologically. And it's the old story. It worked in some, it didn't work in others. So now, you're looking for something that will slow down cytokines. It could be as simple as I've used aspirin and ibuprofen and generic painkillers and different things like that, and blocking co-stimulatory. And so now activating regulatory T cells to slow it down a little bit more, inducing self-tolerance, using vaccines, directing antibodies against self-reactive immune cells. There's a lot of theory in how this goes about. So failure of foreign antigens that resemble self-antigens. Antibodies against foreign antigens cross-react. That's what happens with that strep throat, that streptococcal infection. When we go out treating it, the antibodies basically see markers on lining cells in joints and around the heart valves that are attacked, even though they shouldn't be. Okay. And then a variety of things. Mutations, certainly a part of it. Haptens, okay. Something that gets inside because there's been a damage to a barrier, potentially. All those are things. And I'll stop this here because it's been pretty long, and I don't want to give short shift to this. This is allergies and anaphylactic shock, and I think it's an important unit to spend a little bit of extra time with. So I'll stop it here, and I will continue this with us on Thursday, and I'll probably amplify a little bit on some of the stuff I went to. Thank you very much.